All right, now for the main event, I'd like to hand it over to our speakers. So we have 3 speakers today, all from Janssen and J&J. We have Chandni Valiathan, John Maringwa, and Lixia Pei. Chandni is the head of the model-based meta-analysis group. John is also a director in the MBMA group there, and Lixia Pei is the director of the stats and decision sciences in oncology. And between the 3 of them, we have a healthy mix of statistics and pharmacometrics background. Chandni has a PhD in computational and systems biology. John and Lixia both have PhDs in biostatistics. So with that, Chandni, I'll turn it over to you to begin the presentation.
Great, thanks, Tim. Thanks for that introduction. And while I am pulling up my slides here, I wanted to thank the organizers for giving us this opportunity to present our work here. We're very excited to share what we've been working on. I think it's a very interesting topic and an important one. So I'm really glad for the opportunity to present here. So if we can just confirm that you can see the slides on presenter mode. Yes?
Yeah, we can.
We can see it.
Okay, great. So my name is Chandni Valiathan. I lead the model-based meta-analysis group at Janssen. I'm joined here by John Maringwa, who is also in the MBMA group as a director, and Lixia Pei, who is director of oncology in the stats group. Lixia couldn't be here physically, but she has provided us a recording, and she'll sort of introduce the entire topic for us. And today we're here to talk about surrogate endpoints for accelerated clinical development, and really to share this new method that we've been working on called decision-equivalence metric method. Before we jump right in, I did want to make some acknowledgements. I wanted to thank Paolo Paneke-Galuzio, Merdad Javidi, and Natalia Quijano-Carte, who have been instrumental in this work. So a lot of what you see here is really their effort and ideas and all coming together. So as I mentioned, Lixia was unable to call in, but she has made a recording for us. We'll play it in a bit. She will introduce the topic of surrogate endpoints, the importance of them, why we're interested, and also talk a little bit about how this whole topic has been really great for cross-functional collaboration. I thought, this is our kickoff of a joint webinar, and so I think this is the perfect example for how interesting research can come out of cross-functional collaboration. So with that, I'm going to let Lixia talk to this slide.
My name is Lixia Pei. I'm a director in oncology, statistics, and decision sciences at Janssen. It's my great pleasure to jointly present this work with Chandni and John for evaluating surrogate endpoints for accelerated clinical development. Surrogate endpoint is a measure that relates well with a clinical endpoint of interest in assessing the effect of a new treatment. Surrogate endpoints, if adequately established, can be measured earlier or with less variability, potentially helping to improve trial efficiency in terms of duration and cost. I have been working in hematology oncology, particularly in multiple myeloma trials for multiple compounds. In multiple myeloma, progression-free survival is a well-established primary endpoint. Over the last few years, there are a lot of novel drugs have been developed in multiple myeloma. The median PFS became longer and longer, particularly in earlier line trials. The median PFS ranged from 5 years to 10 years, even for the standard of care. So when we design a trial with novel agents, it's really hard to design a trial with reasonable size and duration. My name is Lixia.
Hi.
So this brings our closely work with Chandni's group because we need to leverage external aggregate level data from multiple trials, multiple compounds to establish the surrogacy, to show the consistency of the relationship between the effect of treatment on the surrogate and the clinical endpoint of interest. This is a great opportunity for our close collaboration at Janssen. Actually, the statistical group and the clinical pharmacology and the pharmacometrics groups have been consistently work closely. So using this as an example, Chandni's group, MBMA, and us have bi-weekly meeting. We gather together to look into the model development, data exploration, and statistical interpretation. Also, we bring in the questions from the clinical team to incorporate the data and the model to see how to address the question efficiently. There are numerous examples of close collaborations for decision making. Now I turn the presentation to Chandni. Thank you.
So as Lixia mentioned, this whole project has been really a very nice opportunity again for our cross-functional collaboration. And it's really very active listening, understanding of how the different groups think and work. And you know, from those type of collaborations, I think some interesting work has come out, and we're happy to share this with you today. So for today's workshop, we will first do a quick review of available methods for surrogate marker selection or assessment. After that, we'll present a motivating example with aggregate level data as well as individual level data by exploring, you know, what's currently available, applying those methods, and really understanding the challenges that we might face or we face often when we're thinking about assessing surrogate markers. After that, I will hand it off to John. He will introduce the decision-equivalence metric method, the DEM method, and he'll also apply the DEM method to the motivating example so we can compare and contrast. And then we'll have a summary and discussion and open it up for questions after that. So first, let's talk about, you know, what have we been doing so far in the field? What are the traditional approaches to surrogate marker assessment? There are many methods already available. We have just summarized here a few that are commonly used or referred to. Prentice criteria, the gold standard, if you may, and a few others related to the Prentice criteria, as well as R-squared, that is, again, most commonly used in trying to understand the relationship between a surrogate marker and a clinical endpoint of interest. So just very briefly going over these common methods, Prentice's criteria is really based on the idea that rejecting the null hypothesis, that is no treatment effect on the surrogate endpoint, implies that you would reject the null hypothesis on the clinical endpoint of interest. And to kind of capture this idea, there are certain criteria that have been defined, and we'll go into some of these details in the next slide. Kind of following up on that is Friedman's proportion explained, which uses some of the equations and ideas from Prentice's criteria to actually think about, you know, say if the surrogate marker does not fully explain a clinical endpoint of interest, then what is that proportion that it does explain? And similarly, this Lee's proportion of treatment effect is something that is independent of Prentice's criteria, but again has that same idea of you know, allowing for the surrogate marker to not really explain all of the clinical endpoint treatment effect, and that way it's a little more forgiving than Prentice's criteria. Then, of course, R-squared, I think we're all familiar with this. It's a measure of variance of the clinical endpoint of interest captured by the surrogate endpoint. And oftentimes when many of these other methods fail, we just look at the R-squared and then go by a high R-squared being sufficient for establishing a surrogate marker. So again, some details on the methodology. So here we have Prentice's criteria. There are 4 criteria usually that we have to check off. The first is about the relationship between treatment and the surrogate endpoint. So that's captured in this equation here. And of course, the coefficient for the treatment should be non-zero, significantly non-zero. The second criterion is about the treatment and its relationship with the clinical endpoint of interest. And so again, this coefficient beta needs to be non-zero, significantly non-zero. The third criterion that needs to be satisfied is the relationship between the surrogate and the clinical endpoint of interest. And again, we should be able to detect this relationship with a gamma coefficient that is, again, significantly non-zero. And I think for most cases, these 3 may be satisfied, but the hardest criterion of all to satisfy is this 4th one, where if you look at both the treatment and the surrogate, Z here being the treatment, S being the surrogate, and how they explain the clinical endpoint of interest, the requirement is that your coefficient for the treatment is 0. That is, the coefficient is not significant, which implies that all of the treatment effect that's being seen in the clinical endpoint of interest is actually explained by the surrogate marker here. So the Friedman proportion explained is kind of an extension of Prentice's criteria and uses the same coefficients and equations, and it's kind of gauging or understanding, say your beta S is not 0. It is a non-zero value with a significance that you have estimated from your data. Then what is that proportion that is explained by the treatment effect, and that kind of also tells you what the surrogate marker is explaining. So that's what this proportion explained boils down to. And then the proportion of treatment effect, again, has this very similar idea, but then it's a new equation. It's only one equation that you fit. There's a treatment term and a surrogate marker term. Again, you're trying to estimate these coefficients, but then you're looking at the proportion of treatment effect coming from the surrogate using a weighted proportion of coefficients. So I know there's a lot of detail here. If you're interested in more discussions, we can go into it later. But this is just a summary of some of the methods that we have explored and looked into that are commonly used to assess surrogate markers. So now introducing the motivating example, we decided to work with diabetes. So we wanted a scenario as we're developing this new methodology, something that is well known in terms of being a surrogate marker for a clinical endpoint of interest. And so one of the most commonly used surrogates, even in clinical practice, is fasting plasma glucose, which is accepted as a surrogate marker for HbA1c. So in this case, HbA1c is a validated clinical endpoint that is used for as the basis for approval of drugs for diabetes, and it itself is a surrogate for microvascular complications of diabetes. But for this example and purpose, we will consider HbA1c as our clinical endpoint of interest. It takes about 3 months for HbA1c changes to stabilize, and so you can imagine how it makes sense to look at an earlier readout of fasting plasma glucose, which takes about 4 weeks or so to stabilize. And again, this fasting plasma glucose test is usually used in the clinic. When you go for your annual checkup, you do a blood draw, and many times you're fasted before and you get your FPG levels checked, and that's a precursor, or it tells you whether you are at risk for developing diabetes. So this is something that's been well established, and also in terms of quantifying that relationship, it was seen in a study of about 7,000 participants that the using fasting plasma glucose resulted in the same classification of 97.7% of US adults in terms of a diagnosis of diabetes, the same as if you would use HbA1c as a marker for classification. So there's a really nice correlation here, really nicely established surrogate marker. So we thought, let's start there where we know what we're working with, and let's see what we get. So the data sets that we used were of two kinds. The first was aggregate level data. The aggregate level data basically comes from a database that was curated using systematic literature review for randomized placebo-controlled phase 3 clinical trials in type 2 diabetes for 4 specific types of compounds. The studies that were included in the analysis reported both HbA1c and fasting plasma glucose at similar time points post 12 weeks. So we were looking at steady state values here. The final set was pretty large. You can see the numbers here, 167 trials, 24 treatments, thousands of participants, and 270 study arms. Individual level data came from one study. It was a phase 3 study for 2 doses of a drug for 26 weeks of treatment. It is a randomized placebo-controlled trial. Subjects were randomized 1:1:1. And again, we looked at HbA1c and FPG values, again at steady state post 12 weeks. So this is just what the data looks like. You can see at the top is all of the data that we have at the aggregate level. The x-axis is change in fasting plasma glucose from baseline. The y-axis is change in HbA1c from baseline. Same thing with the bottom plot, except now the bottom plot shows data from the individual level for that one study. And what you can see is that there is a very nice correlation. It looks very nice, linear, and there's extensive, this data set is pretty huge. So what we decided to do is we wanted to apply some of these traditional methods and see what we got in terms of FPG as a surrogate for HbA1c. And what you'll see is that both at the aggregate level and individual level, Prentice's first 3 criteria is check, 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 easy peasy. But then when you come to the 4th criterion, this is where, again, as I mentioned, that criterion is so stringent, requiring all of the clinical endpoint of interest, the treatment effect there, to be explained by the surrogate. And so this is where things start going not so great. So both aggregate level data and individual data do not pass that 4th criterion. And also, if you look at the Friedman proportion explained, you see pretty low values. So for both of these proportions, the closer you are to 1, the better the surrogate. So being at 0.54, 0.51, 0.59, 0.50 just means that, only about half of the effect that you're seeing at the clinical endpoint is explained by the surrogate, which is not good. Also, if you look at R-squared, which is something that we often fall back on, at the individual level, this R-squared is pretty poor. And at the aggregate level, it is high, but not, usually we would like to see an R-squared of 0.8 or something like that to be really confident about picking a surrogate marker for a trial. So, this is where then I'll hand it over to John, and John will walk through the decision equivalence metric approach. John.
Thanks, Chandni, for the introduction. And can we move on to the next slide? Thanks. So this slide actually talks to the individual level data example that Chandni showed. So here we are just exploring a little bit the data, but within the concept of how we are thinking about this. So in principle, looking at the definition from the Prentice criteria that hovering really around the definition that the test of a null hypothesis under surrogate should be a valid test of no treatment effect also on the true endpoint. So on the left, we just look on HbA1c, so the clinical endpoint of interest or based on observed value. So we have two doses, and we just calculate the observed difference from placebo. So we see it on the first line and then on the third line. But then we also go closely to one of the Prentice criteria where we relate a clinical point of interest to the surrogate. So there we just predict HbA1c from surrogate FPG, and that's the second row and the fourth row. So the idea here is just to show that the observed values and the predicted HbA1c, they tend to point in the same direction. So this is really, if you would be testing hypothesis testing, you would end up with the same conclusion that you have a significantly different relative difference from placebo. So that is the treatment effect, if you will. So on the right-hand side, this is so on HbA1c is exactly the same data that we see on the graph on the left, but here we have the clinical endpoint of interest on top, and but then below FPG is just the surrogate itself. So this is more the data itself, what we say about the differences, the treatment effect based on the surrogate endpoints below, and what we say about the treatment effect based on the clinical endpoint of interest on the plot above. And again, the conclusion is that you tend to point in the same direction, that you would reject the null hypothesis of no treatment effect. In other words, you would identify statistically significant effect based on either the FPG, the surrogate, or HbA1c, the clinical endpoint of interest. So this actually motivates, let's say, the approach here that we are taking with the decision equivalent metric methods. And in summary, this really hinges on, let's say, simulating data, but starting from models that we build based on aggregate level data. And you could also build those models on individual level data. But here we will just dwell on, let's say, models that we build on aggregate level data. So surrogate is a function of treatment effect, one model, and then the clinical endpoint is a function of treatment effect. And when you build these models using the summary level data that we saw, and then we can simulate several trials from this, and we incorporate different levels of variability in the individual variability, model uncertainty. We can incorporate these, and we simulate, for example, 1,000 different trials, and we can vary the specific number of, let's say, study sites. You can have one fixed size, but you can also vary it, so the arm-specific size. And then we do simulations, and for those simulations, we are really looking at in the next step to test the hypothesis of no treatment effects between when looking at the clinical endpoint of interest or when looking at the surrogate. And then we just count the proportion of times when we have the similar decision. So when those decisions match out of those total number of clinical trials that you simulate, because ideally you would normally have a single trial, but then having data from multiple trials actually enables you to put things at the level of probability of having certain outcome, and this is exactly what is being done here. And again, this is really hinging and surrounding the definition of Prentice criteria that a valid null hypothesis with no treatment effect based on the surrogate is also a valid test based on the clinical endpoint of interest. Yes, so this is going a little bit more into detail about how to simulate the data. So there is an application based on the diabetes example that we discussed, and so based on that, on the aggregate level data, as we alluded to, build MBMI models across different treatments and trials, and create functional relationships between the surrogate and treatment and the clinical endpoint of interest and treatment. And then we move on to simulate those individual level data from these models. And one of the key elements here is that we use the predicted surrogate values and clinical endpoints from the model, but then we also the variability. We actually, so this is between subject variability, and this is informed from the literature data that we have, but this is reported measures of precision. So we resample from these. So this is really incorporating this between subject variability, and at the same time incorporating between trial variability, because these are values that come from several trials. And then we simulate 1,000 trials, and here we use a median sample size per arm of 115, which was let's say the median of observed trials in the data set that we used. And we resample from the model uncertainty. And then, as explained before, we just calculate the proportion of trials where the decision made. So this is the hypothesis testing part. We're looking for a statistically significant treatment effect on the clinical endpoint of interest and on the surrogate endpoint. And here with n is equals to 115, we end up with a relatively large agreement score, so the DEM score of 0.99, if you will, 0.988, which is relatively very large. But then we can look into vary this sample size per arm. And actually what we see here is this is the score is actually a function of sample size. As you increase the sample size per arm, the higher, the more you increase the sample size, the higher the score, and which I think makes sense. So there is the threshold, the line that we put pertaining to the value that we get with, let's say, our selected value of n is equals to 115. So essentially, varying the sample size, you can here increase the DEM score, and you see that when you have lower sample sizes, you can also expect lower agreement scores between what you would conclude with the surrogate endpoint and what you would conclude with the clinical endpoint of interest. So this can actually help you to identify an appropriate sample size when designing your study. So now here, what we actually do here is to put the DEM score in perspective of other methods that we just saw. That the relatively low values, like for example, the proportion explained, the Lee method, and also the R-square. But the DEM score here actually provides a more practical and, let's say, general way of looking at data and establishing surrogacy. And what we also see is that it's general enough to capture different model types, and we can also extend this to different types of endpoints, so either continuous, binary, for example, ACR20, in RIA or other applications. And you can also develop the models based on aggregate level data or on individual level data. So when we have these, let's say, traditional methods, not really identifying, let's say, FPG as a surrogate, there may be several reasons. For example, that the correlation, not necessarily very low correlation, does not necessarily mean there's no relationship. So here the FPG actually works on really relating the treatment effect based on the surrogate endpoint and on the clinical endpoint of interest, and then working on agreement between that. So I think with that, I can hand over back to Chandni.
Yeah, thanks, John. So to kind of summarize and put everything together here, what we showed is, we looked at some of the common methods that are often used to assess surrogate markers for clinical endpoints of interest. And we saw that they don't always work, even for scenarios where we think they should. There are certain challenges. We also described the decision equivalence metric method, which really is, the ideas are kind of similar to what has been done before, but then it puts things in a more practical framework. And so when you start talking with the clinical teams and so on, and you talk about hypothesis testing or rejecting the null hypothesis, it's something that they're familiar with. And so it is something that can be understood more broadly, as well as being very practical. So we also saw that, for something where we expected to be successful in identifying FPG as a surrogate for HbA1c using traditional methods, they actually failed. But then with the DEM method, we found results that aligned with clinical observation. So you saw that high, 0.98 value. So the max is 1, right, for equivalence. And that matched what was observed with the 7,000 participants that I showed earlier, reaching about 97.7% of agreement. And so, that gives us confidence that, we're not artificially inflating or finding artificially high scores with this method. And also, through this whole methodology, one thing that a nice sort of corollary, right, that came out, something that showed up in the method, was that we were able to identify an appropriate trial size that would actually help us get a high DEM score. So the DEM score is actually dependent on trial size, which kind of makes sense if you think about it. And so if you think about endpoints that have more variability, you might need higher ends, a higher trial size to actually still have a good surrogate. And so that also plays into, helping make decisions for selecting a surrogate marker. The approach is defined in a general enough way. So John mentioned, the models are general enough. You can use aggregate level data, individual level data, and you can also apply to different types of endpoints. So it's general enough that, you have these knobs to play with and really approach surrogate marker assessment in a very practical way. So with that, I think I've said the last point many times, the DEM method provides a practical, clinical relevant, and generally applicable framework to identify and assess surrogate markers. So that's it. with that, I would like to thank you all for joining and listening, and we can open it up to questions.
Thank you, Chandni and John, for a wonderful presentation. And I have a starter question that I'm curious of. The DEM method, the number's way higher, and I wonder if there's a sense of, is there a threshold at which the DEM method is? It looked like you're using a kind of a hypothesis testing criteria, but is there kind of a benchmark to that where anything under 0.8 maybe is not really good to go forward with? And the corollary to that is, how does that relate to trial success? Do you have a sense of that?
Yeah. So I'll get started, and then John, if you want to jump in, please feel free. So we could go back and check in terms of trial success, but the idea is that it's a probability, right? So you're trying to understand how often your surrogate marker sort of would help you make the same decision as if you're using a clinical endpoint of interest. So ideally, you would want to have it at 100%, in an ideal world, but then you can set the bar, right? Because again, it's probabilities. So it depends on the team. And this is where, again, the collaboration with clinical comes into play, because sometimes they are willing to take a certain level of risk, and that's for the clinical team to decide. But for us, we are able to quantify what that risk is. And I think that is a key point. So again, you need to think about all the other information that you have, the data that you have. I think from my perspective, we look at power calculations and we say, 0.9 is like a really great power. We always have these thresholds that we empirically come up with, and that's the same sort of thinking that we would apply here as well. John, do you want to add anything to that?
No, I think you covered it well. Thanks.
Thank you. That was appreciate the answer. I'm going to go to the Q&A. So the first question is from Bill Denny. In the HbA1c FPG example, the model was for a landmark analysis at 12 weeks. Are adjustments needed if it's going to be performed on longitudinal data or longitudinal analysis?
So it was not on landmark data. We just had a cutoff. So we used all data post 12 weeks. So we just needed everything to have reached steady state. So hopefully that clarifies things. But if you did want to look at data pre-steady state, yes, you would have to incorporate time. John, please go ahead.
Yeah, yes, I just wanted to say, yeah, that's I think that's appropriate. If you would want to include time course, then you would have to change the model, and then you can decide on then when you would want to make this the comparison. So could be, is it early, is it later, but that will be defined by your model. So I would say yes, the model would have to change to incorporate the longitudinal nature.
Yeah, and so just to kind of add, you can see that this modeling step is very general, right? So this model can take any form, and this is where you would start now adding your time factors and things like that. The whole idea here is to really just be able to capture your surrogate and clinical endpoint appropriately.
I think the follow-up from Bill Denny is in John's presentation, he summarized DEM that the model must be developed, simulations are performed, and then decisions are compared. Is this robust to model misspecification?
Well, I don't know, but it's not very dissimilar from any other approach that we do. So we develop models, and then we go through model diagnostics to check how the models capture trends in the data. So, and then we simulate with model uncertainty. So in that case, yeah, I would say when we qualify it to simulate, it has to pass some sort of threshold that it's robust enough for model specification. Of course, you can build many different models, or I don't know. You can even do some sort of kind of model averaging. But I don't know how you then end up simulating from those. But I would say once you do your model development, do diagnostic checks, it should qualify for simulation.
And to kind of add to that, right? So yeah, your results will be as good as your models. And I mean, yeah, we all know this, right? Garbage in, garbage out. I think we've heard it many times. So the idea here, though, is that if you do feel like there is some misspecification, or you don't know, what the right model structure is, there is a simulation step. So you could do sensitivity analysis and try to explore some of those different scenarios and try to understand or get a sense of what the most likely scenario might be. So again, like John said, it's just like any other modeling paradigm. We are limited by the data that we have, the models that we have. So it's not any different in this particular case.
So the next question, and I'm going a little out of order, but are the surrogate and CEI modeled jointly? So can you simulate the trials with correlated noise, or were these endpoints trials simulated independently?
Yeah, I'll take that. Yeah, so I think that's a very good point. So in this application, they are modeled separately, and but yeah, that's a good point to take, that we could model them potentially model them jointly. But in this application, as it is presented here, those models are developed separately. We could join them together, put in a correlation component between those endpoints. So we take that as a good suggestion.
Yeah, and to just add to that, we did in some version of this have dependence of one of the endpoints, with the other. So the equations were not completely independent. But then if you think about it, if you force dependence, then you may end up with a good score as well. So I think you need to think through that a little bit more carefully. So in this particular scenario, just to keep things clean and neat, we did model them independently. But if you do need to consider the codependence, again, in this model development step, you have the ability to do that.
The next one, what would you think would make the DEM identify a surrogate endpoint but not a traditional method? So you mentioned it's practical, but not sure how more accurate is identifying surrogate markers.
Can you repeat that again? Yeah.
So what would you think make the DEM method identify a surrogate endpoint but not the traditional methods? So you mentioned it's practical, but not sure how it's more accurate to identify a surrogate marker.
Yeah. So I think one of the things that we were debating was, why does the DEM score show up as so high when some of the traditional methods failed in this particular scenario? So in this scenario, we know that FPG is a good surrogate. So we know that the DEM method has identified something that the traditional methods have not. And the whole underlying idea here is, I think, much more comprehensive in the way that you look at data and decision making. So if you go back to some of the traditional methods here, right? So the idea, that Prentice started with is about the hypothesis testing. However, when you start looking at these equations, they are formulated in a way that to some extent become very stringent, and you're almost requiring that all of the effect that's seen in the clinical endpoint is captured by the surrogate. With and then also, there is a component of trial size and endpoint variability, but it's kind of not considered directly, but indirectly in these equations. For the DEM method, because you're directly going to the heart of things, which is the hypothesis testing, we are actually exploring multiple parameters in this equivalence of a surrogate for making decisions. So we are considering the sample size, the endpoint variability, and on top of that, the treatment effect. So all in all, it's encompassing multiple factors that are very important in actually practically identifying a surrogate marker. So in this particular scenario, we again identify something that is known to be a good surrogate. I don't know, I think there was a question about how can you know that it's accurate. I think this is half of the answer. There's another part to that answer that I don't know yet. We have to still explore. But one more thing that I will say is that we have seen scenarios where, for example, in the data set itself, R squared is really high, and you would say, oh, that's a great surrogate, let's go ahead with it. But then when we apply the DEM method, we got a really poor score. And so then we try to figure out why is that happening, what's happening there. And we figured out that actually the endpoints was so variable that even if there was a high R squared, if you had a small sample size, your DEM score would be low. And so then we had to have higher sample size to then reach a higher DEM score. And then that gave us more information on actually designing, if you were to use that as a surrogate endpoint. With the R squared, you would just go ahead and kind of do your usual analysis. But with the DEM score, we were able to actually identify how large a trial you actually needed for that surrogate marker to work. So hopefully that has answered that question, maybe not directly, but in different ways.
I know there's quite a few more questions, but we're almost to the top of the hour. So given that this is a joint webinar between the ASA biopharmaceutical section, BIOP, and SXP, I really want to thank Brian Millen, Ted Listig, and really everyone on the executive committee in BIOP for helping us arrange this together. And we look forward to really interacting with BIOP further, and especially working, formally working now as a special working group within BIOP. I think there's a lot of potential for collaboration. So please, if you're an ASA member or a BIOP member, please talk to us at the RISBA if you're going to be attending. And otherwise, please reach out to any of us. So we really want to thank our speakers and thank everyone from BIOP for helping to arrange this joint meeting. So I don't know if Brian, you have anything else to add, or?
No, just thank you to the speakers. Thanks for a great presentation. Thanks all for joining.
Thank you. Thanks for having us. Bye.
Yeah, everyone, and we'll be sending out invites whenever for a future meeting once we have it arranged. So thank you very much.